Also, if you came in late, make sure you're reading 13.1. You are responsible for it because I'm not going to cover it because it is pretty much the anatomy of the respiratory system that you need to understand the content we're going to be in here. Okay, can everybody see my screen with the lungs on it? Yay, thank you, Chloe. Okay. Okay, Anise, I saw your message too. You're good. All right. So we're going to start in 13.2. And we're going to be talking about the overview of what respiration is, how we start ventilating. We're going to get into the physics of air-based, it's all fluids actually, but we're going to think about gaseous-based fluids. We're going to talk about the pressures in the different areas of the respiratory tract. And then we're going to start talking about how those change during the inspiratory-expiratory cycle. And for the next two labs, my online lab's already done the dive response. That's what you're doing this week. Oh, and by the way, for lab this week, you need at least one person who's willing to dunk their face in a little tub of cold water. Hold your breath, dunk your face, and then we're going to be measuring pulse rate and breathing rate. And then we're also going to do some stuff with blood pressure in the extremities. So make sure that you come prepared to have at least one person dunk their face. I would try to have a backup because sometimes people get a bit either claustrophobic or get a little bit anxious if they've had I've had more than one student be like, I've almost drowned a couple of times or once, or I think it only takes once to be afraid of doing this exercise a bit. So you might want to have somebody who is okay with dunking their face and have a backup person too. Okay. Then we're going to talk about the tissues and how compliant they are. And then we're going to talk about some of the lung volumes and capacities that we also do in lab, what minute ventilation is, how the dead space affects respiration, how we actually literally calculate the ventilation rate of the alveoli, and then what the resistance of the airways does to ventilation. So why do you need to breathe? Somebody unmute and tell me why you need to breathe.
To get oxygen into your body and to get carbon dioxide out of your body.
Exactly. And why do we ultimately need oxygen? Where is oxygen?
Respiration.
Yes. And in cellular respiration, aerobic respiration, what is oxygen? So cell bio, molecular bio. You probably did this in micro too if you took micro. Why do we need oxygen in cellular respiration?
It accepts the electrons from the electron transport chain?
Yes. Yes. It's the final electron acceptor from the electron transport chain. And the electron transport chain is using the shift of those electrons to pump hydrogens into that inner mitochondrial membrane, right? So you pump hydrogens into that inner mitochondrial membrane between the two membranes. And then the hydrogen gradient is the thing that runs the pump that makes the ATP, that ATPase pump. You pump 3 hydrogens through, each one kind of does a little twist of the pump, and then eventually you have enough kinetic power to take that ADP and PI and stick them together. So you can't use ATP to make ATP, you use this hydrogen pump. and you pump the hydrogens with the electron transport chain. And if for whatever reason you can't dump those electrons off the end of the electron transport chain, they back up because you can't move an electron into a space that's already filled by an electron. And then you don't pump the hydrogens, and then you don't run the ATPase that makes the ATP, and then you run out of ATP. And that's literally why people die when they don't have enough oxygen. Because if you don't have enough oxygen for enough amount of time, you cannot dump the electrons off into the oxygen, which is the final electron acceptor, and make the water with the hydrogens that are coming through. You cannot run the ATPase because you don't have a hydrogen gradient anymore. You don't make ATP anymore. And since almost everything runs on ATP, everything stops working. Boom, 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 shut down. Okay? And then, as you know, in this whole process of getting the electrons into the NADH and FADH2 that go into the electron transport chain and dump off their electrons, those are the electron transferers, the electron donors, that we're doing the Krebs cycle, right? And then the CO2 is part of what's coming off of the Krebs cycle because we've taken all of the electrons and stripped them, and we're feeding that carbon into it, right? We're taking a glucose molecule, stripping it down, taking a couple of carbons each cycle, sticking them into the Krebs cycle to make the Krebs cycle turn, and we kick out CO2 as a byproduct of making the Krebs cycle turn to get our electrons, to stuff them in the electron transport chain. So we're constantly needing oxygen because we're constantly using ATP, and we're constantly making CO2 as we make ATP. So the whole purpose, as you guys know, and you've known since you were kids, of ventilation then is to get the oxygen in and remove the CO2. But I'm glad we took a minute to take it back to cell bio and that molecular endpoint of our function. So everybody take a big breath in and then take it out. As you ventilate and bring air into the chest cavity, into the lungs, that's bulk flow of the air. And remember, air is a gaseous fluid. So it is a fluid, and it's a gaseous fluid instead of a liquid fluid. And then from there, we get diffusion for the gases into and out of the blood. And then the blood will take them to the tissues through bulk flow, where at the tissues, at the capillary, we're going to get diffusion again. And every time we diffuse, both of the gases go from high concentration to low concentration. And then, as we're going to talk about later, with gases, we also have to consider the pressure that they have. So they will also diffuse from high pressure to low pressure. And most of the time with these two gases, you can think of those kind of synonymously, right? Concentration and pressure work about the same in these two gases in the body. Then it's going to be bulk flow again that takes them via the blood back to the lung surface, diffusion again from high to low concentration that allows you to get the gas CO2 back into the lungs, and then ventilation back out. Okay, so it's really important to understand, since we talk about respiration in layman's terms a little differently than we do in physiological terms, to realize that breathing air in and out is ventilation, and respiration is actually what's happening at the cellular level. And I usually say cellular respiration, but you will need to be specific in your answers to let me know that you know the difference between ventilation and respiration. So, as we were just saying, to get air into the lung, we have to have it flows from a higher pressure to a lower pressure. So to get air into the lung, we have to have a lower pressure in the lung, and you're literally vacuuming the air in. You have to make it a low pressure area, and then air will rush from high pressure to low pressure. John, saw your message. And then when you breathe out, you're doing the reverse. You're pressurizing the air in the lung to make it the higher pressure area. And it's going to have to be higher than the atmospheric pressure. And then you'll be able to get it to flow from high pressure to low pressure. So just as we had in blood vessels, the difference in the pressure divided by the resistance helps you predict the flow, is the how flow is calculated. So all we need to know is the two pressures and the resistance, and we have the flow rate. Okay. You can also make note of the fact that flow is inversely proportional to the resistance, and it is directly proportional to the difference between the two pressure points. So if you have a very big difference in pressure between the two areas, you will have a very high flow rate. If you have high resistance, you will have low flow. So now we're going to think a little bit about the physics of how gases behave in containers. And so Boyle's law is a pretty simple calculation that allows you to predict how if you change the volume of a container with a specified amount of gas in it, it's going to change the pressure. And when we're thinking of pressure, what you need to think about is you need to think about the fact that these gas molecules have kinetic energy. They are banging into each other and transferring that energy in the collisions, and they are banging into the walls of the container. And so the more collisions you have, the more kinetic energy you transfer into the walls, the more pressure you put on the walls. So at a constant temperature, which we are going to assume the lungs are, human body temperature stays pretty constant. If you did this in snakes, you would have to actually do an equation that accounted for it. At a constant temperature, if you change the volume, you're going to change the pressure, and they're inversely proportional to each other. So if we compress a container of gas, we take that same amount of gas and give it less space to bump around it. So you will increase the number of collisions, you'll increase that kinetic energy, and you'll have more collisions on the walls as well. So if you're measuring pressure in that lid, you'll notice that there is now more pressure on the container. And conversely, if we do the opposite, if we decompress it, we have more space for the air molecules to bounce around in. We will have less kinetic energy with each collision, and we will have less pressure. Okay, Deja, saw your message. So this same physics can be applied to the lung, okay? Larger container, molecules are farther apart. We have low pressure. Smaller container, molecules are close together. All right, let's take a minute again to breathe. All right, everybody breathe in. That's your larger container. Now breathe out. That's your smaller container. So our volume change leads to a pressure change. Let's get that settled in our heads real concretely. The first thing that happens is that we have a volume change. That then causes a pressure change. Why am I making a big deal of that? You'll find out in a minute, but let's get that set in our heads. The first thing that happens is a volume change. The next thing that happens is the subsequent pressure change that is caused by the volume change. So the flow of gases will change to equalize the pressure whenever we change the container. And one thing that is important to point out is that always we're thinking about this relative to atmospheric pressure. And what exactly is atmospheric pressure? It's literally the weight of the molecules in the air as they push down and get pulled towards the earth because of gravity. They'll be bounded on the outer parts of our atmosphere by the ozone layer, and they will be more dense towards the earth. So there are literally just fewer of them above you. So if you're at sea level, you have the most molecules of atmosphere above you. If you went to Denver, the Mile High City, you would have many fewer molecules in the air above you. And for the gases that are going to distribute equally throughout the atmosphere, you will have literally fewer of them if you're at a higher atmosphere. And that actually changes your ability to respire well. I was lucky enough during my foreign term at Augustana College to get to go to South America. It was an amazing trip. And in part of the trip, we went to Machu Picchu and we hiked. We hiked from Urubamba up to Machu Picchu. And I had been in Peru for a couple of weeks by that point in time. I got the altitude sickness when I first got there to Arequipa, because Arequipa is pretty high altitude. But then we'd been there, like, I think 2, maybe 3 weeks. We'd done a homestay before we went to Urubamba and started hiking up to Machu Picchu. But then we're hiking, to an elevation of about 14,000 feet. And I felt like at the top of those mountains, I could just see the oxygen molecules floating. Like there's one over there. Let me go get it. So little oxygen at that higher altitude that I was actually like, okay, I'm going to walk for a count of 60, and then I'm going to stop and breathe for a minute. And now I'm going to walk for a count of 50, and I'm going to stop and breathe for a minute. And I did that down to like 20, and then I breathed for like 5 minutes. And meanwhile, the native folks are like zipping past us, like it's nothing, because they are acclimatized to that higher altitude, whereas I was not. So where we're at here in Texas, we are really, really close to sea level. And what we use in all of our calculations is 1 atmosphere, or the 760 millimeters of mercury sea level. And that is something, if you were doing physiology at the University of Boulder, you would have to, and you were doing respiratory physiology and you were doing these finer scale calculations, you would actually have to go in and not assume that you had 1 atmosphere of air. You would have to know where you were at relative to sea level and adjust for that. Okay. All right. So now we can start thinking about 3 main lung pressures. And actually there's 4. I'll get to the end. Transpulmonary is part of transmural pressure, and transmural pressure is pressure across a wall. And we're going to talk about pressure against the chest wall. And what you need to think about, this is sort of a very simplified model of a lung. And each lung, we have the trachea that comes down and splits into 2 main bronchi. And then each lung is wrapped in its own pleura, its own sac that surrounds the lung and has intrapleural fluid in it. So you can either think of this as 1/2 of the respiratory system, or you could think of it as 1 combined lung, because the function is about the same. So however you want to view that in your mind, this would be the main tube to the outside. So the tube, the bronchus, and the trachea. This is going to be our lung in the purple. The green is going to be our intrapleural fluid. The intrapleural fluid is sort of mostly watery, but it's also slippery. It's got some surfactant-like molecules in there. Not surfactant exactly. It's got some molecules in there, glycoproteins, that help make it slippery. Okay. And so what the purpose of the pleura is and the intrapleural fluid is, again, take a deep breath. The lungs inflate a lot and then breathe out. So the lung is going to slide against the chest wall as you inflate it and then slide back as you exhale. And you've been doing this ever since you were born, about 12 times a minute for your entire life. That intrapleural fluid decreases the friction of that lung sliding against the chest wall and allows you to have that slide without any damage to the tissues underneath. Because the lung surface is actually quite delicate, because you want that intimate connection with the capillaries. So both the capillary epithelium and the lung epithelium are just one cell layer thick, super thin. So the diffusion is very easy. But that also means it could be damaged easily if you have any kind of friction. And there is a condition we'll talk about later, pleurisy, where you can get damage if you don't have the proper fluid around it. Okay. And then we have our chest wall. And the chest wall is going to be the diaphragm at the bottom, and then the rib cage, the muscles, and the connective tissues all on the sides and back, bound at the back by the spinal cord, and going all the way up to, everybody find your sternal notch right here. And you can find your clavicles, and kind of right under your clavicles is where the first rib is going to attach into the sternum. And your first set of ribs are they're fibrous connections, not cartilage connections, into the sternum and into the ribs. And they are strong. And they sort of hang the rest of your rib cage like a lampshade. So then ribs and muscle, ribs and muscle, ribs and muscle, all the way down, and then sealed at the bottom with the diaphragm that is this arched muscle that can come down and go up, and it comes down when it contracts. So that's all your chest wall. And when it comes to pressure, there's a little bit more on top of that we have to think about, but we're mostly just going to think about that rib cage and the diaphragm and all of that. So then in those three different areas, we've got atmospheric pressure out here, pressure of the atmosphere. Inside the lung, we're going to have the summed pressure of all the pressure in the alveoli. So the alveoli are those little sac-like, grape-like structures that have that maximum amount of surface area for the gas exchange. And we are going to sum them entirely. All right, so all of the pressures across all of the alveoli is going to be the intrapulmonary pressure, the pressure inside the lung. That's the intrapulmonary pressure, is P alveoli. The intrapleural pressure is here, the pressure that's inside that fluid-filled sac. And then transpulmonary is going to be across the lung wall. And like I said, we're also going to talk about the pressure across the chest wall. So let's dig into these a little bit more. All right. So when the alveolar pressure is lower than the atmospheric pressure, that's when air is going to flow from high to low, and it's going to come into the lung. And you want to think of this as negative pressure breathing. Okay, the thing that brings air into the lung is the vacuum, right? You are a vacuum. When you suck in, you're a vacuum, okay? Remember this, you are a vacuum. When you pressurize that, that's when air is going to flow out, because you're going to have higher pressure in the alveoli than the atmosphere, and it's going to go from high pressure to low pressure. In the intrapleural cavity, it's always 4 millimeters of mercury less at a static point when you're not moving, when you're not in the middle of a respiratory cycle, either an inspiration or an expiration. It's always going to be at least 4 millimeters of mercury less than in the alveolus. Okay. The reason is that the chest wall is holding your rib cage open, right? Your rib cage is holding that thoracic cavity open. When you breathe out, your entire chest cavity doesn't collapse down into nothing, right? That's what it would be like if you didn't have this negative thing going on, that when you breathed out, you would collapse down completely. But you don't do that. When you breathe out, you're making that space smaller, but you're always holding a space open. And that is due into the chest wall, the bones, the muscles, the way they are connected to each other. That is what's going to hold open the thoracic cavity, even as you're pressurizing it. And so then the parietal pleura of that intrapleural sac is attached to the chest wall with connective tissue. In between that is fluid. And then we have the visceral pleura that is attached to the lung surface. And then that fluid is going to pull and have surface tension. And we have both cohesion and adhesion that are going on. We have adhesion of the fluid to the pleural linings on both sides. And then we have cohesion in the middle. And now let's think about another physics property. What is the property of water that allows it to have cohesion and adhesion?
Is it the hydrogen bond?
Hydrogen bonds, right? So this is one place where our liquid fluid is going to be different than our gaseous fluid, because our liquid fluid is mostly water-based, almost all water, with a little bit of other stuff in there, like I said, to make it slippery. And the hydrogen bonds amongst the water molecules are going to have that cohesive force and then adhere to the walls. So as you pull that chest wall out, you pull the parietal pleura out, the water wants to go with it, and the water will pull on the rest of the water. And then that'll pull on the visceral pleura. And then that pulling is going to pull on the lung wall. And that's going to hold the lung wall open. So that negative pressure in there keeps that lung open. Okay? So then what would happen if we didn't have that negative pressure? Boop. This is what a pneumothorax does. So if you ever get a hole poked through the chest wall that allows air to come into that intrapleural space, the air will equalize on the inside and the outside of that lung surface, and you no longer have the vacuum holding it open, and the lung will collapse. And this can happen with a stab wound, a puncture wound, a gunshot wound, and this is also one of the evolutionary benefits of having two lungs that are each in their own pleural sacs. Because if you only get a unilateral puncture wound of a lung, your other lung can stay inflated and can usually ventilate you well enough. Especially being a human or an animal that gets to be cared for by a vet, you can go and get medical services. They will stick a big long needle in here and they will suck this air out, and then the lung will reinflate. And usually, as long as you patch the hole that was originally there, you don't have any lung damage, and you can again start to ventilate the same way you were before you had the puncture. It is deadly if you get bilateral punctures and don't get immediate emergency care, because then you will not be able to ventilate. So if you've ever thought, like, how did that person get shot a whole bunch of times and survive? Well, if they were lucky enough that the bullets did not hit any major vessels, blood vessels, and that they only punctured half of the respiratory system, that is one of the ways that people can actually survive. Plus, the amazing work of emergency healthcare workers, EMS, and doctors and nurses in emergency care. Because if you don't pull that air out and you don't fix the hole, you will not be able to reinflate the lung. It doesn't happen naturally. Okay, so now we know about those two pressures, the pressure in the alveolus and the pressure in the intrapulmonary area. So now we can talk about the pressure differential between the two of them. And that's transpulmonary pressure. So that's the difference in the pressure between the alveolus and the pressure in the intrapulmonary space, in the intrapulmonary cavities. And this relationship is governing the static properties of the lungs. So when the lungs are held open or when they are fully closed, okay? So everybody, again, take a deep breath and hold it. And now let it out and hold it. So at those two places that we held it, that's static properties of the lungs. Okay, so either fully inflated and held, or fully exhaled, fully relaxed and held, or if you just capped off your breath at any point in that respiratory cycle. That's static properties of the lungs, okay? They are also involved in the dynamic properties of lungs, but they'll be constantly changing. And we're going to talk about that in a second. So the transmural pressure then is the pressure all the way across the chest wall. And so, or all the way from the outside of the body to the inner air space of the lungs. So transmural pressure means across a wall. And the pressure outside is atmospheric pressure again. You're in atmospheric pressure. And so you can actually calculate the difference across the chest wall by having the intrapleural pressure minus the atmospheric pressure. And as we were saying, you can get the transmural pressure by going with the pressure inside the alveolus minus the intrapleural pressure, okay? So here are those equations down here. This is table 13.2. And we know that the intrapleural pressure is -4. And then at rest, we've got a pressure in the alveolus of 0. So the transpulmonary pressure difference of 0 minus -4 is going to be 4 millimeters of mercury. Across the chest wall, it's -4 minus 0. And so this is a -4 millimeters of mercury. And what I want you to think about, and these transmural pressures you can actually think about as pressures on the walls themselves. On the wall of the lung, which is that real gentle epithelial tissue, real delicate epithelial tissue with a little bit of connective tissue overlying it. And then chest wall, which, like I said, a lot thicker, muscle, bone, lots of connective tissue. And what we're going to think about is the lung being a balloon-like structure. And you can go and look up these model demos on YouTube, where you take a 2-liter bottle, cut off the bottom, you put like a big wide mouth balloon at the bottom that you can then pull the balloon down. And when you pull it down, you can suck air into the bottle. And when you push it up, you push air out. And then you put another bottle inside that with the top around the mouth of the 2-liter bottle so that the balloon hangs down in the bottle. And again, all you have to do is pull down on that rubbery part at the bottom, and you're going to get that balloon inflating because you've made a vacuum. And then you let it go back up, and then it squishes back out. Okay? So then the transmural pressure is the pressure on the wall of the bottle that would be the chest wall. The bottle is going to be our model for the chest wall. And the pressure, the transpulmonary pressure, is going to be the pressure across the balloon wall. So the inflation of a balloon-like structure requires an increase in the transmural pressure such that PI increases relative to PO. Pressure inside increases relative to pressure outside. Okay, so that pressure across the wall is always going to be positive when we're inflating it. And it's because we're creating a vacuum across it. This is the conceptual part that's a little hard for people to get sometimes. I think it makes sense that we pull on the wall of the chest. That is going to pull on that intrapleural fluid. The intrapleural fluid is going to pull on the lung wall. And as the lung is pulled outwards, we open up the container. And because of Boyle's law, we know that the pressure inside that container is then going down. We made the volume bigger, the pressure goes down. And then if we open up the mouth and attach that to atmosphere, the pressure of the atmosphere is going to be greater than the vacuum, the low pressure we just made inside the lung, and air is going to flow in. But that's happening because we're making the pressure across that transmural pressure bigger because we made the vacuum bigger. Does that make sense? This is one of the places where you really need to listen to me and then go back in the chapter and read this again, okay? Because what we're going to do now is we're going to start putting this together in the inspiratory and expiratory cycles, okay? So these figures in your book, this figure has a lot of information in it. We're going to walk through this flow chart. And then I want you to think about all of the things that are going on in this diagram over here. We're going to start here, go through a cycle, and here. Okay, and this is all inspiration. Up here, you can see that what you've got is your breath volume going up, going down. This over here is a full, it's a full cycle all the way, inspiration, expiration. Here is the pressure in the various areas, okay? So you've got your P alveoli here, you've got your PIP here, you've got your PTP here. So I think it makes sense that we're making that PIP more negative as we breathe in, and then that's helping us bring our air into the lung. This is the part that kind of gets people, is the transmural pressure. Because we're making a bigger vacuum here, we actually increase that transmural pressure. Okay, so let's walk through a whole cycle. All right, and then you can see in your pictures here the different values that we've got. And then something to note, as the elastic recoil force is shown here in this little springy arrow, and the inspiratory muscle force, that of the diaphragm, is shown here in this little red arrow. Okay, so the first thing we do is change the volume of the container, right? And the way to do that in the lungs is to breathe in by contracting the diaphragm. Everybody, breathe in. Diaphragm comes down. If you do a big breath in, you can also lift. You can lift up and out your ribs, and you make that thoracic cavity big. You open the container. The thorax expands. And the first thing, as you pull on that chest wall, you're going to make your PIP more negative. Okay, you're going to make it more negative because you're pulling on it, and you're making that PIP actually bigger for a second as well. You're making this cavity bigger. And so the fluid in there actually gets, the fluid molecules get further apart. The pressure inside the intrapulmonary space gets more negative. Because it's getting more negative, the difference across the lung wall actually gets bigger. And as that gets bigger, as we get this more negative and we open up the lung, as we open up the lung, we reduce the pressure in the alveoli, in the lung, and then we go from higher pressure in the atmosphere to lower pressure in the lung, and air is going to rush in. So I want you guys to remember the container moves first. That changes the volume of the container and then directly affects the pressure inside the container. And then air is going to flow from high pressure to low pressure. I want you to write this somewhere in your notes. Container moves first. That changes the volume, which changes the pressure inside that container in an inversely proportional way. And then fluid is going to move, air is going to move from high to low pressure. If you remember that basic sequence of events, you will not go wrong in this whole process. The reason I keep saying this is because when I ask this question on exams, a lot of times I have people start mixing and matching where these things go, and they get the cause and effect all wrong. And they lose a lot of points because I also don't know that you know what the cause and effect is. So somebody asked it, could I repeat it? Yes, again, container moves first. Pressure inside that container changes second. Third, air moves from high pressure to low pressure. Those are the three principles you need to remember as we start layering on the math and a lot of the physics involved here. If you always go back to Boyle's law and then how a change in pressure dictates flow, you will not go wrong. Container first, pressure second, flow third. So volume of the container moves first, pressure changes second, airflow is third. So we contract the diaphragm and the inspiratory muscles. Let's do it again. We expand the thorax. As we expand the thorax, we pull open that intrapleural space. It pulls along and the pressure goes down in the intrapleural space. Because the pressure goes down in the intrapleural space, the lung then expands. Because the lung expands in that container, then that pressure goes down. Because that pressure then goes down and becomes subatmospheric. So it gets lower than atmospheric pressure, which is 1. Then air will flow from outside to inside the body. And that's inspiration. And again, this is a place where I hope that logically you get to understanding how this flowchart works. But if you cannot, please brute force memorize these steps because I will ask you to reproduce them, sometimes straight from memory, right? Like draw this flowchart and explain it to me kind of thing. Okay. All right. Let's talk about expiration. Expiration is the reverse, and the same principles apply, except here the driving force is that elastic recoil of the tissues of the lungs, because all you're doing with the diaphragm is relaxing it. All right, so everybody take a big breath in. You can feel that diaphragm come down as it contracts. Now relax, just normal breath out. The diaphragm is going to relax back to its dome-shaped relaxed position. It's going to then, the tissues, the elastic proteins in the tissues that were stretched like springs are going to go back to their resting state, and they're going to pull everything back to that resting state. So as you stop contracting the diaphragm and the other inspiratory muscles, your chest wall is going to recoil inward. And then that's going to pressurize first the intrapleural fluid. As you make that container smaller, the pressure goes up. So container smaller, pressure goes up. And then as you have more pressure in that intrapleural space, you're going to put more pressure on the lungs. And then that is going to push on that lung wall and it's also going to have its natural recoil. So those two forces combined are going to make that lung space smaller. As we make the lung space smaller, we pressurize the air inside of it. As that gets greater than the atmospheric pressure, air is going to flow from high pressure to low pressure, and it's going to go out. And again, if we take that back to the math, you can see in the math what happens. All right. We had a really, really negative pressure at the end of our inspiration here in the intrapulmonary space. And we'd gotten back down to 0 pressure in the alveolus as we equalized with the atmospheric pressure. And so we had a big transmural pressure there. And then we relax and we let that recoil come back. And so then we don't have as much of a vacuum anymore. And we're increasing the pressure, right? We were very negative and now we're less negative, less negative. It's still negative because it's being held open by the chest wall, but it's less negative. As this is less negative and this gets pressurized, we have less transpulmonary pressure. And then as this gets pressurized, greater than atmospheric pressure, we're going to have airflow out of the lungs. And it will continue to flow out of the lungs until, again, we equalize the pressure between the alveolus and the atmospheric pressure to where there's no more difference anymore. We go back to an intrapleural pressure of just -4. And our transmural pressure is at its lowest at 4. Go back into the inspiratory cycle. As we contract the diaphragm, we make that intrapleural cavity more negative, right? It's a vacuum, less pressure, more negativity, more vacuum. As we pull on that chest wall or on that lung wall, that transmural pressure across the lung wall gets bigger because we're pulling harder on it. Okay, think of that as that force. We're pulling harder on the wall. And then we're opening the lung and decreasing the pressure inside the lung. When it gets lower than atmospheric pressure, air is going to flow in. Air will continue to flow in until the air in the lung is equalized with the atmospheric air. And that's at a very negative state of intrapleural pressure and our highest pressure across the lung wall. So again, when you think of that transmural pressure, that transpulmonary pressure, think of the actual tension across the lung surface that you're pulling on. When it is its biggest, that's at the fullest part of inspiration. When it's at its lowest, that's at the most relaxed of expiration. Okay, so you can see that in this diagram here, that as we pull on that lung wall, the pressure on the wall itself goes up. And as we relax the pressure on that lung wall, the pressure on the lung wall itself goes down. Does this make sense? Okay, so like I said, I definitely need you to know both the inspiratory cycle and the expiratory cycle, the flow charts, the math, and be able to explain it all. Be able to calculate it if I give you some things, be able to explain it. If I give you some calculations, you could tell me where in the cycle we are at. If I give you some numbers, you could tell me where in the cycle we're at. Yeah? So go read, come back, and I highly recommend you start drawing these out and can do it from memory by the time we get to the exam in a week. Yeah. Come talk to me if you need help with this. All right, so lung compliance then is a factor in these equations because the stretchier your lungs are, the more able they are to do this work. If they are stiffer, they give more resistance and they make it harder for air to flow. Because remember, the more resistance we have, the less flow we have. And this is because we don't get to open the container as much. So when you have normal compliance, you have a normal volume and you have a normal transmural pressure, or transpulmonary pressure, the transmural pressure that is across the lung surface. But if you get decreased compliance, and this can happen through a variety of aging factors or disease factors or disability factors that can come with smoking, when you can get more connective tissue in the lung surface, you get decreased compliance. So you can put more pressure on that lung wall, but you don't get as much volume because it just doesn't have the ability to expand anymore. If you ever had a situation where you have increased compliance, you could get more volume out of it. So our compliance is our change in lung volume over our change in pressure, over our transpulmonary pressure, over that pressure across the lung wall. Oh, great question. Can increased compliance be dangerous or not desired? Yep. There are ways you can, it would be kind of akin to a hyperventilation system or situation, even if you were not having an increased respiratory rate. So yeah, if you took a drug that gave you increased lung compliance, you could hyperventilate yourself again. It would be a similar gas exchange situation to a hyperventilation situation, but you wouldn't increase your ventilation rate. Also, I wouldn't do it because of potential damage to the lung surface itself. It's pretty stretchy, but if you overstretch it, you would rip it. And what you would rip it with, and you could pop holes in your lung, which could be an internal way to get a pneumothorax or an internal way to get air in that intrapleural cavity, pleural effusions. Sorry, that's what those are called. What you would replace it with would be connective tissue. So you'd end up on the other side of the things where you'd end up with decreased lung compliance as the lung fixed itself. Okay, we talked about the stretchability of the lung tissues, but the surface tension at the air-water interfaces within the alveoli also determine lung compliance. So not a big deal for adults or children that have lots of surfactant, but this is a huge deal for preemies that don't have enough surfactant. And we have a synthetic surfactant nowadays that gets injected into the lungs of babies who are preemies who might be having trouble breathing, and greatly reduces surface tension. So lung compliance goes up and they usually don't have many problems. It's why babies can be born as early as like 24 weeks, now even down to 22, 21, super, super early. Half-baked babies is what these guys are nowadays. But if you can get them to about that stage because of the synthetic surfactant, they often can survive. Not always. It's rough for a baby that tiny, but they often can survive because all of a sudden you can reduce that surface tension and they can inflate the lung and they can breathe. And then NICU ventilation has gotten way, way better as well. So without that, then the lung has a very hard time opening up and the compliance is greatly reduced. So surfactant allows for an equalization that doesn't happen as well without surfactant. So there's sort of this compounding factor that when you have alveoli, they're not all exactly the same size. And as we were just saying, on that larger level, air is always going to flow from higher pressure to lower pressure. So when you don't have surfactant, the smaller of the alveoli are going to be more pressurized, and it's going to have a tendency for air to flow into the bigger ones. And then you can get this compounding thing where the smaller ones then collapse, which gives you less surface area for respiration, for gas diffusion. And then you kind of go from bigger to this sort of continues, right? Then in the next ones, you might not be able to hold them open, and the air flows into the bigger ones, and then that one collapses, and you just get less and less and less available surface area for diffusion. But if you have surfactant, the super cool thing about surfactant is that in the smaller alveoli, the surfactant molecules also get pushed closer together. So not only do we have more pressure on the air, but the surfactant molecules actually push closer together. And then they hold open and they don't collapse. And you don't have this difference in them pressurizing the air, because the surfactant molecules are helping to hold that alveoli open. Because as the alveolus tries to close, it pushes their surfactant molecules closer together, and they in return put more pressure on the alveolus to keep it open, and you have more equal distribution of air across all of the alveoli. Okay, so no flow from A to B or from B to A, and you don't collapse, and you keep them open. So this is the reason why surfactant is so important for keeping the lungs open. Please take a look at this calculation. You can see what happens is we can get the area across the, if we took and cross-sectioned it and then went around the perimeter, and we have 2 times the perimeter divided by the radius, is going to give us the pressure inside the alveolus. And then you're just comparing pressures inside the alveolus. And without surfactant, you're going to have greater radius in A than B, and you're going to have less pressure in A than B. But the surfactant actually equalizes the pressure in the two. Okay. Yep. Need to go to class, and we are done with our time. We'll pick up here on Thursday, and we'll go into the rest of the chapter. So anybody who has any questions, I can stick around for a few minutes. If not, have a great start to your week, and I will see you on Thursday.